Hello dear students, in the previous lesson we have talked about fluorescent microscope and different parts of it. For example, we talked about the light source, which is the mercury arch lamp. Then the light coming from the light source passes through the excitation filters that are chosen by the user, and then from the excitation filter the light rays fall on the dichromatic mirror and pass through the emission filter. From the emission filter they can be seen through the detector after passing through the ocular lamp. So now let's see how does this work. Okay, so the principle of to observe the sample through a fluorescence microscope, it should be first labeled with a fluorescent dye or a substance which is known as the fluorophore. मीनिंग अगर आपने फ्लोरेसेंस माइक्रोस्कोप पे किसी सैंपल को देखना है तो यू हैव टू फर्स्ट लेबल इट और स्टेन इट बाय यूजिंग अ फ्लोरेसेंट डाई. यूजुअल माइक्रोस्कोप में आप जो भी स्टेनिंग यूज़ करते हैं उसका अपना एक कलर होता है, जबकि फ्लोरेसेंट डाई डू नॉट हैव एनी कलर एट रूम टेम्परेचर, यानी आपको इसको देखने से कोई कलर नहीं दिखाई देता. But in the microscope you can see the color of the stained organelle or the stained part of the cell. Okay, so the principle is that higher energy light with the shorter wavelength, for example the UV light or the blue light, is generated from the mercury vapor arch lamp. This passes through the excitation filter. This excitation filter is selected by the user. It allows only the short wavelength of the light to pass through and removes all other non-specific unrequired wavelengths of the light. Okay, so now this only selected wavelength of light will pass through this and it will fall on the dichroic filter. Okay, so from the dichroic filter it falls on the sample, and this sample is already labeled with the fluorophore. Now what happens is that the fluorophore absorbs the shorter wavelength of light and it emits the rays of light which are having the longer wavelength. That means that they are lower in energy. Okay, and these wavelengths pass through the emission filter over here. Okay, after striking the sample. So the emission spectrum, meaning the rays of light which are emitted from the object after the dichroic mirror or the dichroic filter, these are usually sharper than the excitation spectrum. And these are of longer wavelength, meaning they have got the lower energy. Okay, so the emission filter blocks or suppresses any of the residual excitation light and passes the desired longer emission wavelength to the detector, meaning now the detector gets only those waves of light which are having the longer wavelength. Thus the microscope will now form the glowing image of the fluorochrome labeled microbe or the cellular structure against the dark background. So the basic point is that the specimen is illuminated with the lights of the specific wavelength that are absorbed by the fluorophores, and this makes the fluorophores to emit the longer wavelength of light or a different color as compared to the absorbed light. So what are the applications and uses of the fluorescence microscope? Fluorescence microscopy is the critical tool for academic and pharmaceutical research, pathology and clinical medicine. This is also used for the demonstration of naturally occurring fluorescent material and the other non-fluorescent substances or microbes after staining with some fluorescent dye. For example, Mycobacterium tuberculosis, amyloid, lipids, elastic fibers, or the different proteins of the cell. It also helps to identify the specific molecules with the help of the fluorescent substances. It helps to trace the location of the specific proteins in the specimen or in the cell. It helps to get the image of the structural component of the small specimen, for example, the cells. Then how then it can also help to locate the DNA and RNA within the cells, which are the genetic material. This also helps to view the specific cells within the larger population with the techniques, for example, FISH, that is fluorescent in situ hybridization. Because some specific cells have got some cellular markers and they can be stained with the markers that can be seen by the help of the fluorescent microscope. It also helps to view different proteins located in different portions of the cell. The fluorescent microscopy allows the researchers to identify various different molecules in targeted specimen or sample at the same time. And that's all for the moment. Thank you.